Okay, so we're going to continue with the honors workshop, and so you can finish filling out your notes. So just kind of as a review, we're going to go through each of them, and you can make sure if there's anything else you wanted to add. So remember the difference between a monomer and a polymer, and that, monomers are going to be a single molecule or compound, like glucose or fructose, amino acid, fatty acid, etc. And then that the polymers are many of those molecules or compounds linked together. Are macromolecules that we're talking about, the carbohydrates, lipids, proteins, and fats, acids, etc. So you should have all of this down already, but remember that the building blocks of proteins are amino acids, that they're typically found in eggs, nuts, beans, meat, vegetables, tofu, etc. There are 52 of those amino acids, and 20 of them are essential to our diet, but 9 of them are not made in our body. And so we have to eat those to get those amino acids. And remember, too, that the peptide bond is the bond that links those amino acids together. Something important to note with proteins is that the structure equals the function. So very important that the structure of a protein or an amino acid is not disrupted, or its function will be disrupted. So you think about babies that have really high temperatures, that can be very bad for their protein development, because proteins will be denatured and then they will be unable to develop correctly, and those proteins will not be able to be produced properly. And so when we think about proteins in our bodies, enzymes are a big protein within our body. So those enzymes are what are going to be speeding up chemical reactions within our body. And so you think about the first enzyme that starts to break down the food that we eat is amylase, which is in our saliva. And so that breaks down the sugars rapidly in your mouth. And so that's why it's important, because they're all throughout our body, and if anything happens to them, then there's other cascades and things that could possibly go wrong. So there are two things that can affect protein function, and that's heat and pH. So just like I said, with high fevers, that's one cause, and then a disruption in pH is another. So you think about the different parts of our body, and our stomach, as you learned in the pH lab, typically with hydrochloric acid has a pH around 3. Our blood has a pH of 7. Our digestive tract has another pH. So it's important to always know what part of the body you're in, and what that pH should look like. So enzymes, again, speed up the chemical reactions in the body, and their structure does equal function. So there's a specific model that we use to describe enzyme function. It's called the lock and key model, and it's the model that's used to describe the function of an enzyme with its substrate. So the substrate is the thing that's going to bind to an enzyme, and it's going to change its shape to speed up a chemical reaction. So you can see here it's a 3-step process, basically. So first, you have an enzyme that has a specific activation or active site for a substrate, and the substrate then going to bind to that active site, and it's going to be an enzyme substrate complex. And then finally, the substrate is going to be released. It's going to be an altered enzyme, but in that process, it sped up some type of chemical reaction. And something to know with this is that the substrate is specific. It only has one place that it can bind to an enzyme. And so that specificity is what makes that lock and key model so important, because it is a very specific process and a specific enzyme and substrate that bind together. So I did say that I'd like you to know this general amino acid structure. So in the center, you have the carbon in the center, and then you have this carboxyl group over here, which is a carbon with a double bond to an oxygen, and an OH group here. And then you have some hydrogens, and then you have an amino group over here, which is a nitrogen with 2 hydrogen groups. Now, remember I said that everything with an amino acid structure is the same apart from this R group over here. The R side chain is going to be different for every amino acid. And like I said, it's the only thing that differs. So kind of know that this is the general structure. And then, if you link more amino acids, then they'll be linked together over here. And then this would be where the peptide bond is. So then if we talk about protein folding, that's kind of where the protein structure equals function back on this slide, and this slide comes into play. So our primary structure is going to be just simple peptide bonds in between amino acids in a long chain like this. And then the secondary is going to be when you create these two different forms of proteins. So they can either be in a beta sheet form or an alpha helix form. And so the beta sheet is just kind of pleated like so, and then the alpha helix kind of reminds you of what a DNA helix would look like, but I drew it kind of like a spring, so it's in kind of a helical formation. And that's the secondary structure. And what causes this to happen is there's hydrogen bonding between different hydrogen groups that causes these shapes. Then we go to this tertiary structure, which is when you combine these alpha and beta sheets, alpha helices and beta sheets in different ways. So you can have two beta sheets together, you can have an alpha helix and a beta sheet, and then those can be combined in different ways. And then we finally get to our quaternary structure, which is when you combine all of these in different ways and you create something complex with it, which is what a functional protein actually looks like. So if you actually search for a quaternary structure of a protein, you get something that looks like this, which is pretty cool. They're pretty amazing, if you ask me. Here's a really good one. So they're humongous, and they have lots of different ways that they are bound together, and they have active sites, like we saw in that lock and key model. And so it shows on here that the red ones are the alpha subunits, and then the green ones are the beta subunits. And so they just create all these amazing shapes, and they're very complex. Like this one has a lot of different complex areas. And so it's very interesting to be able to look at those and see the differences. Okay, so then we haven't talked about carbohydrates yet, so then you'll need to add these to your notes. So the building blocks of carbohydrates are sugars and starches. And there's kind of two different types. There's monosaccharides and polysaccharides. So those monosaccharides are going to be your glucose, fructose, sucrose, those simple sugars. And so simple sugars are going to be things like in table sugar, candy, chocolate, and high fructose corn syrup. It's things that are going to raise your blood sugar very quickly, and then it's going to cause you to crash. So people that maybe have diabetes or something, or hypoglycemia, where they need their blood sugar to rise quickly so they don't pass out or something like that, these are the kind of simple sugars that you'd be looking for, or monosaccharides. And then the polysaccharides are going to be things like starch, cellulose, and glycogen, which are going to be complex sugars. And these are in your potatoes, your pastas, your breads. It's where you link several of the monosaccharides together to get a polysaccharide. Once again, we go back to those stem roots of mono and poly. And, glycogen is the way our body actually stores glucose, stores it in our liver. And then when it's time for us to need an increase in glucose in our blood, our body goes through a process called gluconeogenesis, and it actually takes that glycogen and breaks it down into glucose so our body can use it. This is also why athletes carb load before an athletic event, because it allows them to store more glycogen, so then they have more reserves when they need that during the athletic event, especially like cross country runners. So if you remember in that Plickers game that we did, one of the questions was that our body can easily digest cellulose, which is false. And so